Okay, so for this video, we're going to look at antigens actually present on the red blood cells. So the first one that we're going to talk about is the ABO antigens, ABO blood types. And so basically, there's 6 different genotypes that can be possible. There's so the mom or the dad can both give AA, or it could be AO. It could be BB or BO. It could be OO, and the dad and mom can give AB. Okay, so if it's AA or AO, basically what this person will express is this person will express on their red blood cells type A, and they will have antibodies for anti-B, which will target B antigens. Okay, this person will be type B, and this person will also have anti-A antibodies. This person, OO, will actually just be type O, so it'll just be type O. And this person will be AB, so what you'll see is both A and B. Sorry, type O will actually have both antibodies present. So this will be anti-A and anti-B. And this person, because it has both antigens, it won't have any antibodies for these types. And so these antigens are basically, they're like 7 chains. So type O actually has just the H chain, it's called. Type A, at the end, will have a branch. And type A will have N-acetyl galactosamine, so galacnac it's called. And type B will have a branch of galactose at the end. Galactose at the end. Okay, and so that's how they're kind of differentiated between the 3. Another type of blood type that you'll see is the RH blood type. So with RH, the one that's actually, there's different kinds of RH, but the one that's most specifically recognized is RHD. And so say you have a mom that is RHD negative. So if it's RHD negative, she does not have the antigen for RHD. And if the father is RHD positive, when the mom becomes pregnant, you can actually have the infant, which will also be RHD positive. Okay, so the blood in the infant, if the baby is Rh positive, it'll have antigens for Rh on their blood. And if this blood gets out of the placenta and mixes with the mother's blood, the mom can produce antibodies for anti-Rh. And then these antibodies can actually go in, and if they cross the placenta, they'll go in and they'll attack the fetal blood, and this can cause an abortion. So in these cases, if you know that the mom is Rh negative and the fetus is Rh positive, you'll have to give the mom some kind of immunosuppressant to prevent this from actually happening. So that's the Rh. I've got a couple other ones listed here, like Kells, Duffy, Kidd. They basically all have the same concept in which they produce a type of antigen that are not present in everybody in the population. So when you give a blood transfusion, they can cause some kind of hemolytic anemia and reaction. Okay, so now to test for these antibodies and antigens, there's 2 kinds of tests. There is the DAT test, which is the direct antiglobulin test, and the IAT test, which is the indirect antiglobulin test. So in the direct antiglobulin test, basically what you do is you are going to, in a test tube, you'll take somebody's blood, and you'll wash the blood, so that the plasma, so you remove all the free-floating immunoglobulins, and what you're left with is just the red blood cells in solution. That's all you want, just the red blood cells from the individual. And if these red blood cells have any antibodies attached to them, so if there's this is to test for autoimmune hemolytic anemia, autoimmune hemolytic anemia. So it's basically to test whether there is antibodies attacking the red blood cells, and that's what's causing the anemia. So if there are any antiglobulins present on these, so any antibodies present on the red blood cells, what you do is you add polyspecific IgM, that basically this IgM will target immunoglobulins. So you add these IgMs, so you basically add it to the solution, and what will happen is if there are these immunoglobulins present, this IgM will go in and actually bind, and it'll cause a clumping, and so that'll be a positive test for DAT. So then you know that there are antiglobulins present on the red blood cells. After that, you can use monospecific IgM, basically to test which type of immunoglobulin or complement protein is present on these red blood cells. Okay. You can also do this at warm and cold temperatures. Basically, at warm temperatures, you're testing whether immunoglobulins are present, and if you do it at cold temperatures, around 4 degrees, IgM targets more the complement system, so it's more specific for the complement test. With the indirect immunoglobulin test, what you are actually doing is you take the person's blood, and in this case, you're actually removing the red blood cells, and you're left with just the plasma. And for this one, you're testing whether the person has immunoglobulins against a specific antigen. So you remove the red blood cells, and what you're left with is all the person's antibodies in their blood. Okay? And then what you're going to do is you're going to draw blood from other individuals that you know are positive for different antigens that you want to test for. So say here we have the assay, and in this test you'll have the different individuals' bloods that you want to test for. So say this person's Rh positive, so that would be testing whether there's the so if you add the blood here and it clumps, that would be positive test for anti-Rh. So then you know that this person has antibodies to test for Rh. And you can also test for type A, you can test type B, and wherever they clump, it actually shows a positive test for the indirect antiglobulin test, and it'll show that the person has antiglobulins for it. One other thing to note, actually, I forgot to mention with the direct antigen test, is you want to make sure that there's not cross-reaction. You want to make sure that there's not mixed field reaction. So in this case, say half the blood actually clumps. You see half the blood clumps, or a portion of it clumps, a portion of it doesn't. In this case, it could be indicative of a blood transfusion in which the person's actually making antibodies against the blood transfusion that has been added to the person. So if the person was type B and you've added type A and this blood transfusion was type A, the person would be producing anti-A against it and actually causing, and that's why these antiglobulins would be on here. And so it wouldn't actually be an autoimmune attack, it would be an attack against the blood that has been transfused, and in this case, what you'll see is some of them will show positive, but then there'll be a lot of other blood that is actually not clumping. And so that's called a mixed field reaction.